convert succinyl CoA to succinate. Okay? Thiokinase. Succinyl CoA thiokinase. اوكي it doesn't matter اذا مكتوب كده معناته انا غلطان. طيب succinyl CoA converted to succinate. Then the succinate is converted into fumarate through enzyme succinate dehydrogenase. And here is the first reaction to produce fat H. The first are last because there is only one fat H is produced in the citric acid cycle. So then the fumarate is through a fumarase enzyme. It's hydrolysis, so it adds water to convert fumarate into malate. Then the malate is converted to oxaloacetate by malate dehydrogenase. And this is the third and the last reaction which produces NADH. So through this cycle, we regenerate oxaloacetate again, which will condensate again with other molecules of acetyl CoA. And in three different reactions, we got NADH, so produce 3 NADH, 1 fat H, and 1 GTP at substrate level. So the total will be 12 ATP. 3 multiplied by 3 plus 2 for the fat H plus GTP, so 12. So per acetyl CoA, we get 12. But per 2 acetyl CoA, which is coming from 1 glucose, we get 24. In the question, read carefully. If I ask you how many ATP is produced per molecule of acetyl CoA in citric acid cycle, the answer will be 12. If I ask you how many ATP is produced per molecule of glucose by citric acid cycle, 24. If I ask you what is the total production of energy per molecule of glucose in the presence of oxygen and mitochondria, that means you have to calculate the glycolysis, you have to calculate the pyruvate dehydrogenase complex and citric acid cycle. 38. If I said what is the net production? 36. Okay? So today we will talk about gluconeogenesis. For gluconeogenesis, as we mentioned before, there is tissue in the body, they solely dependent of glucose. They cannot live without glucose. Okay? That means the body have to produce glucose irrespective of your fed status. If you ate something, or if you fast, or if you starve, the body have to produce glucose. If you get glucose from the food or not. Why? To make those tissue survive. Those tissue mainly the brain, and we said because there is 2 sources of energy, either fat or carbohydrate. Most of the fat, they cannot cross the blood-brain barrier, so they cannot get access to the brain. That's why brain need to have the energy from glucose. The RBCs, because they lack the mitochondria, so they cannot oxidize fat, but they can use the anaerobic glycolysis. And the kidney also, part of the kidney, and also exercising muscle. We said during exercise, the muscle, contracting muscle, they compress the blood vessel, reduce the supply of blood to the tissue. So it deprives tissues from oxygen. So that means they can only rely on the anaerobic glycolysis. So all this tissue need glucose. The glycogen, if you remember, the storage form of glucose, Okay, if you fast, the liver start to produce glucose from glycogen. Okay, but if the fasting period prolonged more than 12 hours or 18 hours, all the glycogen stored in the liver will be finished. So it can only, the glycogen in the liver can only maintain the blood glucose for 12 to 18 hours. What after 18 hours? What will happen to the brain, RBCs, and kidney? They will start to produce glucose from non-carbohydrate sources. And the process of producing glucose from non-carbohydrate sources is called gluconeogenesis. So gluconeogenesis is the production of glucose from non-carbohydrate sources. تفضل. بدون كربوهيدرات.
لكن في اكسجين.
ده موضوع تاني. لازم يكون في اكسجين ولازم يكون في ميتوكوندريا. To produce glucose you need both. You need oxygen and you need mitochondria. طيب. So what are the non-carbohydrate products that can be converted to glucose? They are the lactate. You remember lactate? The lactate from the muscle can be transported to the through the blood to the liver, and there it can be converted to glucose. Also the glycerol. We will know more about glycerol when in the upcoming lecture of lipid metabolism, and also amino acid and alpha keto acid in general. So what will happen? Fasting, the glucose will come from glycogen. If the fasting prolonged more than 18 hours, then the body will start to produce glucose from non-carbohydrate, lactate and glycerol and amino acid. If there is starvation, أيوة. Gluconeogenesis, yes.
هذا في النورمال هيومن بودي ولا حنتكلم عن abnormal؟
لا بنتكلم في النورمال. كيف abnormal؟ This is normal human body, but not all people get starved. Okay. فاللي بيحصل في البرولونج يعني لما تحصل مجاعة what will happen؟ هيحصل starvation because this is not fasting لأنه there is no food. So the body will start to produce glucose from other, and that's why, like when, because we cannot store amino acid. So what will happen? The body start to destroy the tissue, the muscle, and that's why the people with malnutrition or starvation, they are wasted. They become wasted. They become very thin because the destruction from the muscle to get amino acid, and then the amino acid converted to glucose. So during the overnight fasting, during a fasting overnight, 90% of this process, the production of glucose occur in the liver, and 10% in the kidney. But during starvation, في المجاعة, kidney become the major glucose producing organ. Okay, as we said, the main source of production of glucose from non-carbohydrate are glycerol, lactate, alpha keto acid, and amino acid. Those are the main substrate for gluconeogenesis. طيب, gluconeogenesis is closely resembles the reversal pathway of glycolysis. If you remember, in the glycolysis, we start from glucose, we end by pyruvate. If we want to produce glucose, we need just to reverse. We go back from pyruvate to glucose. But there is a problem here, because if you remember, and I repeated it 100 times, there are 10 reactions in glycolysis. 7 reversible, 3 irreversible. So if you want to reverse the whole pathway from pyruvate to glucose, you have to bypass those 3 irreversible steps, because the reversible step, it will there will be no problem because it can go in both direction. But the irreversible reaction need to get bypassed by different reaction by different enzyme. So 3 out of 10 reaction of glycolysis are irreversible. The 7 reactions, the rest 7 reactions are common for both glycolysis and gluconeogenesis. So it is just a reversal of glycolysis from pyruvate to glucose, but with exception in the 3 irreversible reactions. Type, if you remember, the irreversible reaction was the first reaction, glucose to glucose 6-phosphate by hexokinase or glucokinase. This was the first irreversible reaction. The second was the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate by phosphofructokinase 1. The third was a conversion of phosphoenol pyruvate to pyruvate by pyruvate kinase. So those are the 3 irreversible reactions in glycolysis that we need to bypass them to produce glucose from pyruvate. So conversion of pyruvate to phosphoenol pyruvate by pyruvate. So those bypassed here in gluconeogenesis. First, we want from pyruvate to go to phosphoenol pyruvate. To do this, we need 2 enzymes actually, not only 1. The first one is the pyruvate carboxylase, and the second one is phosphoenol pyruvate carboxykinase. This is the only tricky part, because the second one, you want to convert fructose 1,6-bisphosphate to fructose 6-phosphate. Usually, if you want to remove phosphate, the enzyme name is phosphatase, because you are removing phosphate. So here this will be fructose 1,6-bisphosphatase. So it works on fructose 1,6-bisphosphate, and since it removes one phosphate, to produce fructose 6-phosphate, so we call it fructose 1,6-bisphosphatase. The other one is to convert glucose 6-phosphate to glucose, and it is the same. Since it works on glucose 6-phosphate, and since it just removes the phosphate group, we call it glucose 6-phosphatase. So what happened in details? First, conversion of pyruvate to phosphoenol pyruvate. So this is the first irreversible step. So this enzyme pyruvate carboxylase converts pyruvate to oxaloacetate. If you remember, pyruvate had 3 carbon and oxaloacetate have 4 carbon. So if we want to convert pyruvate to oxaloacetate, we need to add 1 carbon. So it is carboxylation reaction. Any carboxylation reaction in the body need the biotin. So this is the coenzyme is needed for any carboxylation reaction, because it bound to the carbon dioxide and add it to the products. So at the first step, we convert pyruvate to oxaloacetate. Okay? And oxaloacetate synthesized in the mitochondria has to enter the cytosol, but due to membrane impermeability, the oxaloacetate cannot leave the mitochondria. There is no transporter to take the oxaloacetate from inside the mitochondria to the cytosol. To solve this problem, we need to convert oxaloacetate back to malate by malate dehydrogenase. Malate can cross the inner mitochondrial membrane into the cytosol. So transport to cytosol, where it is reconverted again by cytosolic malate dehydrogenase. So you need two malate dehydrogenase, one in the mitochondria and one in the cytosol. The one in the mitochondria will convert the oxaloacetate to malate. Then the malate will cross the membrane to the cytosol. And then you will convert it back to oxaloacetate. And then oxaloacetate under decarboxylation reaction with the phosphoenol pyruvate carboxykinase will be converted to phosphoenol pyruvate. And then through the reversible reaction until we reach fructose 1,6-bisphosphate, and by the action of fructose 1,6-bisphosphatase, this will be converted to fructose 6-phosphate. Reversible reaction until glucose 6-phosphate. Glucose 6-phosphate will be converted into glucose by glucose 6-phosphatase, and then the free glucose will be released to circulation or to the blood or outside the liver or the kidney. So here is the conversion of pyruvate to phosphoenol pyruvate. You need biotin because it bound carbon dioxide. You need ATP in this reaction. So pyruvate, by addition of carbon dioxide, will be converted to oxaloacetate. This occurs inside the mitochondria. Oxaloacetate cannot penetrate the membrane, so it should be converted to malate by the malate dehydrogenase inside the mitochondria. The malate, when it reaches the cytosol, converted back to oxaloacetate. And then by phosphoenol pyruvate carboxykinase, oxaloacetate with decarboxylation reaction is converted to phosphoenol pyruvate. So here we reached the glycolysis pathway, or one of the intermediate of glycolysis. So what we need is just to reverse the pathway, and we start with the pyruvate inside the mitochondria, and we end with phosphoenol pyruvate. And then reversible reaction until we reach fructose 1,6-bisphosphate with bisphosphatase, fructose 6-phosphate, pack glucose 6-phosphate with glucose 6-phosphatase, free glucose released into circulation. So the two enzyme that convert pyruvate to oxaloacetate and oxaloacetate to malate and malate to phosphoenol pyruvate is just to bypass this. So pyruvate is converted to phosphoenol pyruvate. Reversible reaction, reversible until here. Fructose 1,6-bisphosphate. Here we will need phosphatase. Fructose 6-phosphate. This is reversible. Here we will need another phosphatase, glucose 6-phosphatase. We will get free glucose. So, as I said before, to convert fructose 1,6-bisphosphate to fructose 6-phosphate, the second irreversible reaction, you need fructose 1,6-bisphosphatase. So this will release phosphate group, and then you will get fructose 6-phosphate. This is an important regulatory site of gluconeogenesis. When we talk about the regulation of glycolysis, we said that all the irreversible reactions, they are very important regulatory sites for glycolysis. It is the same for gluconeogenesis. Those are the sites where, because I mean, the body cannot do glycolysis and gluconeogenesis at the same time. This will be just kind of stupidity. Unfortunately, the cell is not stupid. So either the pathway is directed through glycolysis or to gluconeogenesis. And to control this, the site or the rate limiting steps are the irreversible reactions. So here, fructose 1,6-bisphosphate to fructose 6-phosphate by fructose 1,6-bisphosphatase. The last irreversible reaction, we convert glucose 6-phosphate to free glucose, D-glucose, by adding water, release this phosphate group, and then it will, under the action of glucose 6-phosphatase. If you remember from the glycolysis lecture, I told you that the function of phosphorylation is to trap the glucose inside the cell. You remember? Yes. So if you remove the phosphate, then glucose will no longer become trapped. So it will goes outside. It will follow the normal osmosis, the normal osmotic, from high concentration to low concentration. If you release 3, 4, 5, 6 glucose, they will go outside, because outside there is less glucose, because it's just normal osmotic. Through GLUT2 receptors, because the gluconeogenesis is mainly happen in the liver, especially in prolonged fasting. The liver have transporter GLUT2, which is bidirectional. It means it just depends on if you have high level of glucose outside, they will diffuse inside. If we are in a glycolysis phase, this glucose will be phosphorylated and trapped inside. If we are in a fasting state with high glucagon level and we are in gluconeogenesis, the glucose 6-phosphate will be converted to glucose, and the amount of glucose inside will develop, become more and more. Outside there is less glucose, so the glucose will diffuse through GLUT2 outside. The gluconeogenesis is a very costly process. It costs a lot of ATP for the body to produce glucose from non-carbohydrate. To maintain normal blood glucose level in the circulation for the brain and RBCs and the other tissue that is solely dependent of glucose. And since brain is very important, the cell consumes a lot of energy to produce and provide glucose to the brain during fasting. So here we started from pyruvate and we converted pyruvate to oxaloacetate. For 1 molecule of glucose, we will need 2 molecules of pyruvate. So we will need 2 ATP here just to convert pyruvate to oxaloacetate. And then we will need 2 GTP to convert oxaloacetate to phosphoenol pyruvate. And then we will need 2 ATP here to convert 2 3-phosphoglycerate molecules into 2 1,3-bisphosphoglycerate. And we will need 2 NADH here to convert 1,3-bisphosphoglycerate into glyceraldehyde 3-phosphate. So in total, to produce 1 molecule of glucose, we need 2 molecules of pyruvate, 4 ATP, 2 GTP, plus 2 NADH. So 2 pyruvate in addition to 12 ATP. Just to produce 1 molecule of glucose from 2 pyruvate molecules. So it is very costly process, since it is very important to maintain normal blood glucose level for tissue that is solely dependent on glucose. So how those two processes are regulated? They are regulated by hormones. During fed state, when you eat, your body releases insulin. And from the previous lecture, we know insulin activates glycolysis. If you are eating, we don't need to produce glucose because glucose is coming from the diet. But we need to store the glucose for the fasting period. So during fed state or the postprandial state, the insulin activates the glycolysis. During fasting, when the level of blood glucose starts to drop, the body don't release insulin, but instead body release glucagon. And glucagon released with when the level of glucose is reduced. So it will work first on glycogen. If glycogen is depleted, it will work in gluconeogenesis. So this is the hormonal regulation of glycolysis. Fed state, insulin, glycolysis, glycogen synthesis. Fasting state, glycogenolysis and gluconeogenesis. We start first with glycogenolysis. We degrade the stored glycogen, which can provide glucose for up to 12 to 18 hours. After that, we start to produce the glucose from non-carbohydrate. The source are lactate, like it is anaerobic, the end product of anaerobic glycolysis, the lactate. So for example, the RBCs, they continuously produce lactate because all glycolysis in RBCs is anaerobic. So this is the major source. So all the lactate will go to the liver, will be easily converted to pyruvate, and the pyruvate convert to glucose. So from RBCs we get lactate, goes to the liver, and from the liver this lactate is converted to glucose, which go back to RBCs, and this is called Curie cycle. Like the liver provide the RBCs with glucose, the RBCs produce lactate, and lactate goes to the liver, and liver produce glucose again and send it back to RBCs. انيروبيكلي بس في السيتوسول. هي ما عندها ميتوكوندريا. لان عشان عمليه النقل تحتاج ميتوكوندريا. No no I mean lactate, for the lactate you don't need anything. قلنا يا اخوانا ال RBCs they don't have mitochondria. That mean all the glycolysis will be anaerobic. If there is oxygen or there is no oxygen, because there is no mitochondria. So all the glycolysis in the RBCs, the end product is lactate. This lactate will go back to the liver and reused to produce glucose, especially during fasting or starvation. واضحة؟ Any question? No question? Thank you very much. هو كومبلكس لانه هو فيه more than one enzyme. هو فيه 3 enzyme في ده بس. عليكم السلام تفضل. السلام عليكم. Good afternoon. انتوا جروب A صح؟ اكيد. اوكي. طيب. So today we will continue discussing different metabolic pathways, and today we're gonna discuss gluconeogenesis. And before we start to talk about gluconeogenesis, we will go very briefly and revise the citric acid cycle. So, as we said, citric acid cycle or tricarboxylic acid cycle or Krebs cycle is the most important metabolic pathway for producing energy, because actually citric acid cycle provides 65 to 70% of the total energy produced by cells. That's why it's the most important metabolic pathway for energy production. And we said that it takes place in the mitochondria. So all the enzymes of citric acid cycle are located within the mitochondrial matrix. And it starts with a condensation reaction between oxaloacetate and acetyl coenzyme A to produce citrate. We talked also about the connection between glycolysis or the aerobic glycolysis and citric acid cycle. Okay? And the connection is the final product of aerobic glycolysis is the pyruvate. So the pyruvate produced in the cytosol first have to get inside the mitochondria. Inside the mitochondria, under the action of pyruvate dehydrogenase complex, this pyruvate is converted to acetyl coenzyme A. This reaction, the conversion of pyruvate to acetyl coenzyme A, first, it's not belong to citric acid cycle or to glycolysis. It is an intermediate reaction that just connect the final product of aerobic glycolysis to the first reactant of the citric acid cycle, the acetyl-CoA. The most important about this reaction that this enzyme complex have 5 coenzymes and 3 enzymes. The 5 coenzymes are thiamine, vitamin B1, and lipoic acid, and coenzyme A, fat, and NAD. So 5 different coenzymes. So pyruvate get inside the mitochondria, converted to acetyl-CoA. And the reaction is oxidative decarboxylation, because pyruvate have 3 carbon, is converted to acetyl-CoA, which have 2 carbons. Okay? So inside the mitochondria, the acetyl-CoA condensate with oxaloacetate. Oxaloacetate 4 carbon and acetyl-CoA 2 carbon. They produce citrate 6 carbon. By the action of enzyme called citrate synthase. Then citrate convert into isocitrate, reversible reaction, and the enzyme called aconitase. After that, isocitrate with oxidative decarboxylation reaction is converted to alpha ketoglutarate by action of enzyme called isocitrate dehydrogenase. So isocitrate have 6 carbon. With this decarboxylation reaction, which remove 1 carbon dioxide, it gives alpha ketoglutarate, which have 5 carbon. This reaction, the isocitrate dehydrogenase reaction, is the first reaction that produce NADH. So the first NADH is produced upon conversion of isocitrate into alpha ketoglutarate by the action of enzyme isotrate dehydrogenase. Then the alpha ketoglutarate is converted to succinyl CoA by action of enzyme called alpha ketoglutarate dehydrogenase. This gives the second NADH. It's also decarboxylation reaction. Okay? So the alpha ketoglutarate was 5 carbon. When we remove carbon dioxide, it will be 4. So succinyl CoA have 4 carbon. And the second reaction to produce NADH. Succinyl CoA converted to succinate by enzyme called succinyl CoA thiokinase. This reaction is the first reaction to produce GTP at substrate level. So we convert succinyl CoA to succinate by succinyl CoA thiokinase. Then the succinyl CoA is converted to fumarate by succinate, convert to fumarate by succinate dehydrogenase. This reaction is the first reaction to produce fat H. So succinate to fumarate by succinate dehydrogenase give fat H. Then the fumarate by hydrolase reaction, enzyme called fumarase, it give malate. The malate is converted to oxaloacetate, which is the last reaction, by action of enzyme called malate dehydrogenase. And this gives the final and the last NADH. So through the cycle, we completely oxidize acetyl CoA and produce 2 carbon dioxide and regenerate oxaloacetate, which condensate again with other molecule of acetyl CoA for other cycle. During this cycle, we will have 3 NADH, which equivalent to 9 ATP, 1 fat H, which equivalent to 2 ATP, and 1 GTP. So in total, by 1 molecule of acetyl coenzyme A, we get 12 ATP. But by 1 molecule of glucose in citric acid cycle, we get 24. But if I ask you the total production of energy in the aerobic condition, in the presence of oxygen in the cell, so the total amount of ATP will be 24. But what about glycolysis? Glycolysis 8. What about converting pyruvate to acetyl CoA? طيب نحن قلنا per molecule of glucose citric acid cycle will give 24. متفقين؟ By converting pyruvate to acetyl CoA, we get how many NADH? 1. But we will have 2 pyruvate. So how many NADH? 2. How many ATP? 6. And from glycolysis? 8. 8 plus 6. 8 plus 6. ده حساب كمان يعني. This is mathematics. It's not biochemistry. 14 plus 24. 38. This is the total production of energy in the presence of oxygen and mitochondria in the cell per molecule of glucose. But if I ask you the net production of energy in presence of oxygen and mitochondria in the cell per molecule of glucose, 36 بالضبط 36, because we lose 2 in the investment phase of glycolysis. Repeat which part? طيب very quickly. Citric acid cycle 24. متفقين؟ Pyruvate to acetyl CoA 6 ATP. في حدا معترض؟ Pyruvate to acetyl CoA 6 ATP. في أي واحد معترض؟ Glycolysis 8 ATP. متفقين؟ 8 plus 6 plus 24. 8 plus 6 plus 24. This is the total number of energy. 38. If the question was the net mean you invest and you get the profit. How much? 36, because there is 2 ATP in the glycolysis in the investment phase. Okay? Okay, so today we will talk about gluconeogenesis. I told you in the previous lectures that there are tissues that solely dependent of glucose as a source of energy. They cannot survive without glucose. And they are very important tissue. The brain is important, I guess. RBCs, kidney, and exercising muscle. طيب نحن قلنا انه main source of energy in the body either fat or glucose. The fat cannot cross the blood-brain barrier. That's why only the glucose can be used to provide energy to the brain. The RBCs, they don't have mitochondria. That means they cannot oxidize fat. They cannot use fat as source of energy. That's why they totally dependent on glucose. Also, part of the kidney, they only depend on glucose, and exercising muscle. In exercising muscle, when the muscle contract, they compress the blood vessels. So the blood will not flow to the tissue. There will not be any oxygen. It will be only anaerobic condition. And in this anaerobic condition, only glucose can be used for anaerobic glycolysis. You cannot oxidize fat without oxygen. So these tissue, they always need glucose. طيب, when we eat, the glucose will be available. It will come from the digestion of carbohydrate, which flow through the portal vein to the liver. So everything will be fine. But what if we don't eat? If we fast, what will happen to those tissue? Brain, RBCs, kidney, and exercising muscle. What will happen to them?
They will break fat.
No, they will break glycogen. They cannot use fat. Those tissue, they can never use fat. So what will happen? The liver will start to degrade the glycogen. And you know glycogen is polymer of glucose. So we will have a lot of glucose that will go to circulation, and all this tissue will be happy. But any store have a limit. And the limit of glycogen in the liver is up to 12 to 18 hours. And usually you don't fast more than this. So usually 12 to 18 hours, and then you feed and you get feed. But what will happen if the fasting continue more than 18 hours? Glycogen finished, خلص, no glycogen. What will happen for this tissue? They need glucose always. They don't understand that there is no glucose and you are not eating. So the body can produce glucose from non-carbohydrate sources. This process is called gluconeogenesis. It is from the name. Genesis means synthesis. Glucose, new. So producing new glucose from non-carbohydrate, because if you are not eating, there is no carbohydrate, and the glycogen is finished. So this is the term gluconeogenesis came from this. So what are the non-carbohydrate sources that we can use to produce glucose? They are glycerol. We will know glycerol more when we talk about lipid metabolism. Lactate is produced from anaerobic glycolysis. For example, in RBCs, it can be reused by the liver to produce glucose. Alpha keto acid, an amino acid. Okay, so this special pathway, the gluconeogenesis, require both mitochondria and cytosol. You have to understand this, because we said the glycolysis take place in cytosol. Citric acid cycle take place in the mitochondria. Gluconeogenesis post mitochondria and cytosol. You need the post compartment to produce glucose. So during overnight fasting, 90% of this gluconeogenesis occur in the liver and 10% occur in the kidney. But what happens if the fasting is prolonged, 2 days, 3 days, 4 days? Even we don't call it fasting anymore. We call it starvation. Starvation. Fasting هو الصيام العادي. Starvation في المجاعات. No source of food. أو أنت ما لقيت أي food for more than 2-3 days. This is not fasting. This is starving. Okay. So during this is prolonged fasting or the starvation, the process is take place in the kidney. Okay. طيب معناتها most important organ for gluconeogenesis is the liver in the prolonged fasting. لو في starvation يكون الكدني. Okay. So let us have a close look to the gluconeogenesis. If you remember in glycolysis, I was just keep repeating that the irreversible reactions are very important. There are 3 irreversible reactions in the glycolysis, and those 3 irreversible reactions are the most important reactions because they are the rate limiting steps, because they are the regulatory step of glycolysis. Because I was focusing on that because those 3 reactions are the only irreversible reaction in glycolysis. In gluconeogenesis, we want to produce glucose, so we want to reverse the pathway. We want to go from pyruvate back to glucose. Those are 10 reactions. 7 out of 10 are reversible, so there is no problem. They can go in post direction. But 3 is irreversible. So those 1 need to be bypassed. If you want to convert pyruvate to glucose, you need to find a way for the irreversible reaction. So it will be new enzymes and new reaction to bypass these irreversible steps. So, as we said, gluconeogenesis closely resembles the reverse pathway of glycolysis. 3 out of 10 reactions of glycolysis are irreversible. The first reaction, when we convert glucose to glucose 6-phosphate, is irreversible. The third reaction, when we convert fructose 6-phosphate to fructose 1,6-bisphosphate by phosphoroctokinase 1, is irreversible. And the last reaction, when we convert phosphoenol pyruvate to pyruvate by pyruvate kinase, is irreversible. Those 3 reactions, we need to find a solution to them. So the 3 irreversible steps of glycolysis are catalyzed by the enzyme glucose to glucose 6-phosphate. It is hexokinase or glucokinase. Fructose 6-phosphate to fructose 1,6-bisphosphate is the phosphoroctokinase. Phosphoenol pyruvate to pyruvate is pyruvate kinase. So these 3 stages are bypassed. They should be bypassed by alternative enzyme specific to gluconeogenesis. So these 3 enzymes are specific to glycolysis. There are other enzymes to bypass those, and those new enzymes are specific to gluconeogenesis. So first, if we want to convert pyruvate back to phosphoenol pyruvate, we will need 2 enzymes, pyruvate carboxylase and phosphoenol pyruvate carboxykinase. This is the tricky step in gluconeogenesis. The other 2 steps are straightforward. If you want to convert fructose 1,6-phosphate to fructose 6-phosphate, you need to remove 1 phosphate. Fructose 1,6-bisphosphate, you want to convert it to fructose 6-phosphate. You need to remove phosphate group from carbon 1. The enzyme for dephosphorylation or for removing phosphate usually called phosphatase. The name is the name of the reactant, fructose 1,6-bisphosphatase, because it removes the phosphate. The same for converting glucose 6-phosphate to glucose. You want to remove the phosphate. The name is the name of the reactant. It is dephosphorylation reaction, so it is phosphatase. So the enzyme name is glucose 6-phosphatase, which convert glucose 6-phosphate to free glucose. Convert glucose 6-phosphate to free glucose. If you remember, I hope you remember, I said the function, like in glycolysis, the first step, when we convert glucose to glucose 6-phosphate, I told you that this reaction is important for trapping the glucose. You need to convert glucose to glucose 6-phosphate to get trapped inside the cell. Here is the opposite. You remove the phosphate, the glucose will be free. And then the glucose will decide depending on the osmosis. It will go from the area of higher concentration to area of lower concentration. So if you have a lot of glucose inside and the amount of glucose outside is low, so glucose will diffuse back outside the cell. If the glucose outside is more than inside, it will go inside. But if you phosphorylate the glucose, it's end. It cannot go, even if the osmotic effect is toward outside. طيب. The first step, to reverse, first step we need to convert pyruvate to phosphoenol pyruvate through oxaloacetate. But the aim is to convert pyruvate to phosphoenol pyruvate. To do this, we need two different reactions. First, we convert pyruvate to oxaloacetate. How many carbon in pyruvate we have? 3. Oxaloacetate? 4. So if we want to convert pyruvate to oxaloacetate, we need extra carbon in form of carbon dioxide. And for each carboxylation reaction in the body, you need biotin. So biotin carries this carbon, add it to pyruvate, and convert the pyruvate to oxaloacetate. From 3 carbon pyruvate, it will become 4 carbon oxaloacetate. This happens inside the mitochondria. The problem with oxaloacetate that it cannot cross the membrane of mitochondria to go to the cytosol. And that's why we need to convert oxaloacetate to malate. The malate have the ability to cross the mitochondria, so it will go outside into the cytosol. And we convert it back to oxaloacetate. And then we convert oxaloacetate to phosphoenol pyruvate by enzyme called phosphoenol pyruvate carboxykinase. Here is the reactions. So here we have the pyruvate inside the mitochondria. 3 carbon. To become 4 carbon oxaloacetate, we need carbon dioxide. And the carbon dioxide, to add the carbon dioxide to pyruvate, we need this coenzyme biotin. So biotin carry the carbon dioxide and add it to pyruvate. Then pyruvate converted to oxaloacetate. Oxaloacetate cannot cross the mitochondrial membrane, so it will convert to malate by malate dehydrogenase. The malate will come outside the mitochondria into the cytosol. We convert it back to oxaloacetate by cytosolic malate dehydrogenase. And then oxaloacetate will be converted to phosphoenol pyruvate by action of enzyme phosphoenol pyruvate carboxykinase. And phosphoenol pyruvate can continue in the reverse glycolysis pathway to produce, yes, until fructose 1,6, because all others are reversible reaction. So we convert pyruvate to oxaloacetate by adding carbon dioxide using this coenzyme. Oxaloacetate cannot cross the mitochondrial membrane. It will be converted to malate. Malate can cross the mitochondrial membrane, go out to the cytosol, convert it back to oxaloacetate. oxaloacetate will convert to phosphoenol pyruvate. So from here to get here, we needed two reactions, and we need to even convert oxaloacetate to malate to get from inside the mitochondria to the cytosol. All the other reaction from phosphoenol pyruvate to 2-phosphoglycerate is reversible reaction, so no problem here. From 2-phosphoglycerate to 3-phosphoglycerate, reversible reaction, no problem. From 3-phosphoglycerate to 1,3-bisphosphoglycerate, reversible reaction. Here, reversible reaction until we reach fructose 1,6-bisphosphate. From fructose 1,6-bisphosphate to fructose 6-phosphate, this is irreversible reaction. That means we need to bypass this step. And we bypass it by the enzyme fructose 1,6-bisphosphatase. It is dephosphorylation. You need just to remove the phosphate group in carbon 1 by adding water to convert fructose 1,6-phosphate to fructose 6-phosphate. And this reaction is important regulatory reaction for gluconeogenesis because it is one of the reactions that bypass the irreversible reaction of glycolysis. So by the action of fructose 1,6-bisphosphatase, we get fructose 6-phosphate. This is the second irreversible reaction. Fructose 6-phosphate to glucose 6-phosphate, no problem. It is reversible reaction because they are glucose 6-phosphate and fructose 6-phosphate are isomers. So it is isomerase. From glucose 6-phosphate to glucose is irreversible reaction. So we need to remove this phosphate. And the enzyme will be glucose 6-phosphatase. So we need just to remove this phosphate group by adding water. The name of the enzyme is the name of the reactant, glucose 6-phosphate, but since it's dephosphorylation reaction, we call it glucose 6-phosphatase. So we will convert glucose 6-phosphate to free glucose. And the free glucose, then this process will build up more and more glucose inside the cytosol. Outside, since it is a fasting and the level of glucose will be low, so the glucose will diffuse from cytosol to outside the cell in the circulation. This process takes place mainly in the liver, as we said. So it will happen through transporter, the GLUT2, which is bidirectional. During fed state, it can allow the glucose get inside. During fasting state, it allows glucose to go outside because it is a function of the liver to maintain the normal blood glucose level. So the process of gluconeogenesis is very costly process. The cell pay a lot of ATP to produce glucose from pyruvate because, as we said, to maintain the blood glucose for those tissue, the brain, RBCs, and the kidney is very, very important. And that's why the cell is willing to pay a lot of ATP for this. So if we look here, to convert pyruvate to oxaloacetate, we need 1 ATP. For 1 molecule of glucose, we will need 2 pyruvate to 2 oxaloacetate, so 2 ATP in this step. 2 GTP from oxaloacetate to phosphoenol pyruvate. 2 ATP from 3-phosphoglycerate to 1,3. And even 2 NADH from 1,3-bisphosphoglycerate to glyceraldehyde. So for the cell just to produce 1 molecule of glucose, the cell needs 2 pyruvate, 4 ATP, 2 GTP, and 2 NADH. So it means that the cell needs around 12 ATP and 2 pyruvate molecule to produce 1 molecule of glucose for those cells. How the body regulates this process, the gluconeogenesis? As we mentioned in glycolysis. The hormones in the body, they are responsible for regulating this process. The cell cannot do glycolysis and gluconeogenesis in the same time, because it will be just stupid, and cell is not stupid. So at one time, either you have glycolysis or you have gluconeogenesis. Both pathways cannot coexist at the same time. So what regulates this? What makes the decision that the cell should be in a glycolysis state or it should be in gluconeogenesis state? It is your fed state. When we eat, our body produces insulin. So it is the anabolic hormone. Insulin, as we learned from the glycolysis lecture, is an activator of glycolysis. So whenever you have insulin, that means you are in a fed state. That means you have abundance of glucose, and we need to store this glucose. 7 سنوات انعجاف و7 سنوات السمان. Okay? During fed state, you need to store the glucose in form of glycogen. This process is regulated by insulin. So what will happen if you fast? No feeding, no diet. The glucose level, because during feeding or postprandially, you have high glucose in the blood. And insulin will handle this glucose and will store part of this glucose. When you don't eat, the glucose level will go down. So there will not be any insulin released. But other hormone called glucagon will be released. So in the fasting state, you will have low blood level of glucose and you will have high level of glucagon. Both insulin and glucagon, they are released from the pancreas. So during fasting state, that means the body needs glucose. Glucagon will be released from the pancreas. The first function of glucagon is to destroy the glycogen. Glycogen is polymer of glucose. So it will destroy glycogen, produce glucose, which will be released into circulation. But any storage have a capacity. After 12 or 18 hours, it will finish. يخلص خلاص ما في. So the glucagon after 18 hours, after depletion of all glycogen, will produce the glucose from non-carbohydrate product, which are lactate, glycerol, amino acid, whatever. Okay? So glucagon will activate the opposite pathway, the gluconeogenesis, to produce the glucose from non-carbohydrates. The question is, from where we will get this source of non-carbohydrate? First, lactate. We have a lot of RBCs in our body. They always work anaerobically. So RBCs in our body is always producing lactate. This lactate will go to the liver. The liver will convert the lactate to pyruvate and then to glucose, and will release the glucose back to the RBCs. RBCs will anaerobically use the glucose, produce lactate, send it back to the liver, and this is called Cori cycle. Continuous interaction between liver and RBCs. RBCs always produce lactate, liver take lactate and produce glucose, send back to the RBCs. Okay? What will happen during starvation if this continue for very long time? There is no food. The body will start to destroy the muscle, the proteins, to get amino acid. And amino acid will be destroyed to get pyruvate, and pyruvate will be converted to glucose. That's why during starvation or prolonged malnutrition, there will be a body wasting. Okay? They lose a weight and they become very thin because they use their own protein and their own muscle to maintain the glucose level for those important tissue, the brain and the RBCs. عندك سؤال انت؟ واضح؟ في اي واحد عنده اي سؤال؟ ها؟ الجليسرول من وين؟ سؤال ممتاز. نحن لما ناخد الليبيد ميتابوليزم ان شاء الله هنعرف from where we get glycerol. لكن for your own sake from adipose tissue. Yes. from adipose tissue. اوكي؟ في اي واحد عنده اي سؤال؟ انتوا مالكم الليله يعني ما يعني كده هادين كده كويسين انتوا؟ مش جروب ايه ولا انا غلطان؟ خير؟ اه اه
process of eating our proteins
Mhm.
and
degrading our protein you mean. Mhm.
how long does it take to start?
يعني if you don't eat for example 3 days, so the glycogen will finish and then the lactate and will not be enough, so it will start like after 3 days it will start. But for how long you can survive without eating? 3 days بس؟ No. This is the homework. نشوف المرة الجاية how long you can survive without eating. 3 days? No, it's not 3 days.
I have noticed for 2 days.
Okay, next lecture we will see. Thank you very much. If you don't have question, thank you. And thank you for your behavior today. I'm really glad.